The cell membrane part 2, phospholipid bilayer. Now whereabouts is the phospholipid bilayer present in a cell? Now it can be present, number 1, in the cell membrane. Number 2, the mitochondria, the outer and the inner membrane are phospholipid bilayer. In the chloroplast, the outer membrane, the inner membrane and the thylakoid membrane all are phospholipid bilayers. Also the lamellae are phospholipid bilayers. The nuclear membrane is a double phospholipid bilayer. So the nuclear membrane is a double phospholipid bilayer, meaning that there is one layer and then there is another layer. So there are two layers. And then the Golgi membrane, the RER membrane and lysosomes are membrane bound vesicles. So they would be also made of the phospholipid bilayer. Now the role of phospholipids in the phospholipid bilayer, there are 3 points which need to be considered. Number 1, the fatty acid chains can be saturated or unsaturated. You know, saturated fats are those which are animal fats and they solidify. But the unsaturated fats like olive oil do not solidify. So if they do not solidify, we have more unsaturated the fatty acids, the more fluid, because they are bent, they fit together more loosely. The second point is the length, the tail length. So the tail length is very, very important. The tail length is very important. The longer the tail, so more carbon to carbon bonds. So longer the tail, less fluid. Now please understand that the cell membrane must remain fluid in order to the cell to survive and be living. If the cell membrane solidifies, the cell will die. Now as we decrease the temperature, the cell membrane becomes less fluid. So these are 3 points that we need to consider. Number 1, are they made up of saturated or unsaturated fatty acids? Number 2, the tail length. And number 3, as the temperature decreases, it becomes less fluid. So please pause the video and have a look through it, and please try to remember these 3 points. Now coming to the role of cholesterol. Now the cholesterol also has a hydrophilic head. It also has a hydrophilic head and it also has a hydrophilic tail. So that is why it is situated in between the phosphate heads and the tails, because it must have the same properties. Then the animal cells have cholesterol and phospholipids in the same amount. So I mean, if there are 100 of those, there are 100 of those. In plants there is less cholesterol. In prokaryotes there is no cholesterol. In prokaryotes, which means in bacteria, there is no cholesterol. Prokaryotes are bacteria. And the cholesterol gives it mechanical stability. So another point that you have to remember, you need to pause the video and remember all these points. So basically cholesterol maintains and regulates fluidity. So the fact that you have a regulator on your fan regulates the speed of the fan. So what do I mean by the word maintains and regulates fluidity? So this is what you have to understand, and this is what has to be understood in this part of the chapter. Now we come to the roles of phospholipids. Now the phospholipids are of 2 types. The first role that we talk about is the saturated and the unsaturated fatty acids. Now important thing which we need to understand is the more the unsaturated. Please remember, unsaturated fats are those which are plant origin, and you know, olive oil, corn oil, they remain fluid at all times. So if you have more unsaturated, there will be more fluid, because as they are bent, you see the phospholipid molecule has these straight 2 chains, but if this is unsaturated, it is bent, and one could be straight and one could be bent. So more fluid as being bent, they fit together more loosely. So more unsaturated is the ones which don't have C, they have a double bond between C, and they have all not, in saturated is all C and C are single bonds. Then we have talked about number 2, the tail length. The tail length is important because as we look at it, the longer the tail length, less fluid the membrane. And number 3, as the temperature decreases, it becomes less fluid. Then we come to the role of glycolipids. Glycolipids are the one which are the carbohydrate attached to the phospholipid bilayer. Then the glycoproteins are the ones which are attached, the carbohydrate chains, to the protein. And then proteins, as such, we have a huge number of proteins in the cell membrane. Number one is the receptor molecule. Glycolipids and glycoproteins act as receptor molecules. You see, how does insulin know that it's got to work in the liver cells? So, the receptor molecules for hormones. So, if you look at the molecule insulin, it has a certain shape. Now, on the liver cell, there would be receptor molecules where this would fit, and that would be a glycolipid or a glycoprotein. So the insulin receptor in the liver cells are glycolipids and glycoproteins. Then we have glycolipids and glycoproteins are cell markers, or we say antigens. Antigens are surface markings, like your Face ID, like your phone would only open when you look at it. So you have a Face ID. So similarly, every cell, every human being has a specific antigen on every cell of their body. And that antigen could be of any shape. So there's an antigen this shape, then this one has an antigen this shape, this one has an antigen this shape. So we have different antigens on the surface of cells. Even bacteria has different antigens. So certain bacteria have a specific shape, and then of course we study that in immunity, how that results in a response being generated. Then we come to proteins, and we discuss a very important protein, a transport protein or a channel protein. Now if you look at the structure of this protein, as you know, they're made up of amino acids. Now another thing which you have to remember from chapter 2 is amino acids are of 20 different types, and they have 20 different R groups, and each R group is responsible for certain properties. So there are R groups which are hydrophilic, and there are R groups which are hydrophobic. Now if you look at the arrangement which I have drawn here, inside a cell membrane, when a protein is present, so the blue part, now this would have the hydrophilic R groups. Hydrophilic R groups, why? Because ions have to pass through this. Could be potassium ion, could be sodium ion, could be chloride ion. So the blue part is the area, the protein which has hydrophilic R groups. The amino acids would have hydrophilic R groups. While the part which is in between, now this area, which I am shading in grey, this area is the one which is in between the tails. Now this area would have the hydrophobic R groups, because please understand, it's between the tails, and the tails are hydrophobic. This area is the hydrophobic part of the phospholipid bilayer. So another thing which you have to understand is the structure of the protein molecules which are fixed in between the cell membrane. So the hydrophilic R groups and the hydrophobic R groups. Please pause the video and have a look at this and make your notes so that you can remember this, because we have a lot of exam questions on this. The next topic is cell signaling, and cell signaling is basically to maintain homeostasis or to transmit one signal to another part of the body. So what we have is, we have signaling molecules, and signaling molecules are basically called ligands. Ligands are special small molecules, but they are water soluble. That's why they cannot pass through the hydrophobic part of the cell membrane. So what we have is, we have protein receptors on specific cells, like insulin only works on the liver cells, glucagon only works on the liver cells. So they will be receptors for those signaling molecules on specific organs. So the signaling molecule first gets attached to a protein receptor. Now the shape of the protein receptor is complementary to the signaling molecule. So it gets attached to it, and it results in a change in the shape of the protein receptor, and that is called transduction. That is called transduction because the shape of this changes. The shape of this molecule changes. And this results in activating the G protein. So G protein acts like a switch. And then this G protein activates this enzyme. And then this enzyme produces certain secretions or certain enzymes, and those further activate other enzymes, and which further activate more enzymes, and that will result in the production of either a certain chemical or something which is needed by the body. Now there are different names which I want to talk about. Number one is transduction, in which the shape of this protein is going to change. Then another word which we use is amplification. Amplification is this whole thing in which it is amplified. And another word that we use is signal cascade. So all these words must be understood. They are very, very difficult biological English, but we need to be understanding this. So transduction, amplification, and signal cascade. And the cell signaling ensures that the body is working in response to the environmental changes. It's working. Like for instance, if you eat a lot of sugary stuff, then the blood sugar is going to rise and the hormone insulin is going to be produced, which is going to result in the glucose being stored in the liver cells as glycogen. So that has to be normalized through these signaling molecules which are called ligand. Now, how do we calculate the surface area and volume of a 1 centimeter cube? A cube has 6 sides. So, if it's 1 into 1 is equal to 1, but it has 6 sides. So, the surface area will be 6 centimeter square. The volume of this 1 cm cube would be 1 into 1 into 1. So this will be 1 cm cube. Volume is length into breadth into width. So 1 cm cube. Now the surface area to volume ratio will be 6:1. Now similarly, if you look at the 2 centimeter cube, now the surface area of this will be 2 into 2, 4. And then this 4 into 6. So the surface area will be 24 centimeter square. This is 2, this is 2. So 2 into 2 is 4, and then you multiply it by 6 sides, so it's 24 centimeter square. Now the volume of this will be 2 into 2 into 2. So that will be 8 cm cube. Now the surface area to volume ratio will be 24:8. Now we can simplify this, and this will be 3:1. So what has happened when the size has increased? What has happened when the size is increased? The surface area to volume ratio has reduced. Here it was 6:1, now it is 3:1. So this is what you have to understand. And as the size of a cube increases, the surface area to volume ratio decreases. The surface area to volume ratio decreases. Please pause this and understand this. We have to study the effect of animal cell, and for an animal cell I've chosen a red blood cell. So a red blood cell, when it is placed inside pure water, now a pure water would have no solutes in it. So what will happen is that water will enter the red blood cell and the cell will swell up and burst. So the red blood cell will swell up and burst. Now the same red blood cell, if you placed in a solution which has the same concentration as the cytoplasm of the red blood cell, then of course there is going to be, now look at the words that we use. We're going to say there's going to be no net movement. We say there will be no net movement. Please remember, water will always move. But when we say there will be no net movement, it means 10 water will move in and 10 will move out. Now the cell will remain normal. Nothing will happen. It won't shrink, it won't swell up, it won't burst. But if I place the red blood cell in a concentrated solution, then what is going to happen is water is going to move out and the red blood cell is going to shrink. Because what is happening is that if we have a concentrated solution outside, that means it has a lower water potential, and inside the red blood cell, the cytoplasm has a higher water potential, so the water is going to move out. Please understand, the water is going to move out by osmosis. So the red blood cell is going to shrink. Now in plant cells it's a different phenomena, because if you place plant cells in water, now the water will move into the plant cell and it will become fully turgid. But if you place the plant cell in a concentrated solution, then water is going to move out. And the water molecules, when they move out, this results in the cell membrane. The cell membrane is going to shrink away from the cell wall. So you can see the cell membrane in both the cells is very differently placed. Now these areas where it is attached is actually the areas where there is plasmodesmata. Plasmodesmata. And the cell membrane is now leaving, it's no longer very closely adhering with the cell wall, and it is leaving the cell wall and it is coming to the center of the cell. So as the protoplasm continues to shrink, it begins to pull away from the cell wall, and this process is called plasmolysis, and a cell in which it has happened is said to be plasmolysed. The cell is said to be plasmolysed. Another word which we use here is incipient plasmolysis, and that is the point at which plasmolysis is about to occur is referred to as incipient plasmolysis. Now let's talk about measuring water potential. Water potential can be measured in pressure units called kilopascals, and that of course we write as kPa, but the whole thing is kilopascals. Now because pure water has free water molecules and it has no solute in it, so it we say has zero. Water potential is measured in 0 kilopascals. Now whenever you add a solute, it becomes negative. So this cell A is minus 250 kilopascals, cell B is minus 400. So I had asked you to remember it like there's 250 grams of sugar in it, or 400 grams of sugar in it, or 110 grams of sugar in it. Now you can understand how the movement is going to take place, because if we say cell A is more negative than cell C and cell C is less negative than cell B and cell A, now where would there be more free water molecules? Now you've got to understand is that C has 110, so it has say 110 grams of sugar in it, so it's got the least. So movement will be from C to B or even C to A. And then of course, because this is 250 and this is 400, cell A, so it will also be from cell A to cell B. So please understand how the movement of water molecules take place, how we represent this. Now we show water potential as the letter psi, and this is how we write it, and this is how we represent it in different MCQs that you will get. So minus 110, minus 400, minus 250. So we can say, we can word this as cell A is less negative than cell B, but cell A is more negative than cell C. So this word less negative and more negative you've got to understand, because we might not give you figures, we might just write that in the question, and for that you need to interpret that. So just give it some figures, -10, -20, -30, something like that, to understand the question. The practical activity that we need to do, you need to consider about 6 practicals which you need to be discussing and theoretically doing and thinking about it. Number 1 is diffusion using whisking tubing. Number 2 is diffusion using plant tissue, and you can use pieces of beetroot. Number 3 is effect of size on diffusion. So you can be given pieces of potato and you can be asked to cut them up and use them in different sizes to see the effect of size on diffusion. And number 4, you need to study osmosis in plant tissue. For that you need to study 2 practicals. Number 1 is observe osmosis in plant cells. And number 2 is determine the water potential of a plant tissue. Determine the water potential of a plant tissue. So you need to be doing these about 5, 3 and 2 of these. So 5 practicals which you need to study, and you need to understand the theory behind it. And then of course you have a lot of questions from paper 3 which have come on this topic. So you need to go through those. And that will be the end for the chapter on cell membrane. And I will try to get you to make a video on the practical activity as soon as I can. Thank you so much.